Hi everyone, welcome to my channel. Today's topic is neuromuscular junction. If you want this topic in Hindi, you can visit my Physiology Hacks channel for Hindi video. Okay. So neuromuscular junction. In this video, we'll be talking about not only junction but all the events happening at neuromuscular junction. So that comes under neuromuscular transmission. Okay. So you can get a question, a long question in which you can have a describe neuromuscular junction and explain the events at this junction, right? As neuromuscular transmission. So you'll be starting with what is neuromuscular junction, the introductory part, and then you will skip to the transmission part, right? So how you are going to describe the answer. So let's see that. So neuromuscular junction. What is neuromuscular junction? It is basically specialized synapse between motor neuron and skeletal muscle. So this is a motor neuron which is supplying this muscle fiber, and here the nerve terminal is basically having synapse with this muscle fiber. So this synapse is called neuromuscular junction. So how you are going to describe this junction? So you have to draw an image in which we have synaptic terminal and some part of our muscle, right? Fine, this structure. So how you are going to label it? This is part of nerve terminal or synaptic knob, we can say. So we'll write it, this is a presynaptic terminal or presynaptic membrane. Since this is synapse between a motor nerve and skeletal muscle, so this is a synapse. So before synapse, the membrane is called presynaptic membrane. After synapse, this membrane is called postsynaptic membrane, right? Fine. So we have this terminal buttoned synaptic knob in which we have vesicles present over here. We have neurotransmitter packed in these vesicles, right? We have calcium channels on this terminal, which is a presynaptic terminal. Okay, the space between both the membrane is called synaptic cleft. We have some dimension of this synaptic cleft. So this is basically the muscle membrane is basically having foldings, fine, so that we can have more surface area and more receptors at this level. This is called motor end plate. Fine. On this motor end plate, we have some channels present over this surface, which are nicotinic based acetylcholine channels. Okay, and below these are some channels which are sodium voltage gated sodium channels. We'll see how these calcium channels, these acetylcholine receptors and sodium channel work for transmission of some impulse from a motor nerve to muscle. Okay, so that is neuromuscular transmission. So this image you will draw if you are asked only about neuromuscular junction. Okay, so this is part of neuromuscular junction which you will be explaining. So then you can start the transmission part. So let's see what we are going to write in transmission. So we can start that neuromuscular transmission have two parts: events at presynaptic terminal and events at postsynaptic terminal. So let's first see events at presynaptic terminal. So first of all, action potential arrives in the nerve fiber, okay, because we have a motor nerve ending. So obviously that motor nerve have some impulse from our cortex or any supraspinal areas, right? Since we are talking about any motor activity here, this is basically motor nerve is supplying skeletal muscle, right? So we can do some movements. For example, I'm moving my fingers. So those movements are basically having corticospinal pathway. So from cortex, there is a command to move these fingers like this. So we have impulse from various areas, supraspinal areas, and the signal comes in that motor nerve. Okay. Now motor nerve have action potential arrived near the terminal. Okay. So this is presynaptic membrane. Since we are going to describe now the events at presynaptic membrane only, so at neuron level only, right? At nerve level. So action potential is coming in the nerve toward the terminal, right? So what happens? This is called depolarization of nerve, right? So what happens? This action potential, it basically opens the voltage gated calcium channel. Since this action potential is nothing but a voltage change, and whenever some threshold voltage is achieved, these calcium channel open up. Okay, so which lead to calcium entry into this nerve terminal. Okay, whenever this happen, there is basically the vesicles are present over here, and they are going to fuse near the membrane, and ultimately the neurotransmitter which is present inside these vesicles is basically released here. And now this neurotransmitter is nothing but acetylcholine. Okay, so you can describe these events like this. So opening of voltage gated calcium channel occur, then calcium enters into the membrane, then fusion of synaptic vesicles occur, and acetylcholine is released, which we call exocytosis of neurotransmitters. Fine. Once this happens, the potassium channel open up and repolarization occur, which closes the voltage gated calcium channel again. So these are the events which occur at presynaptic membrane. But this is not a way to present, okay? So because this attaching the vesicles toward the membrane and releasing the neurotransmitter, it's a big process. So we have to describe these events in detail. So let's go through all these events happening at this level, how basically these vesicles are formed and then attached to the membrane and released in the neurotransmitter released in the space. So let's see in somehow detail about this pathway, right? So first of all, how this acetylcholine is synthesized and packed into the vesicle, we should know. So acetylcholine synthesis. So what happens basically? Choline enters with the help of some transporter which is CHT. So choline transporters, right? Choline transporter basically transfer the choline inside the nerve terminal, and we have acetyl-CoA from mitochondria. Mitochondria are present in the nerve itself, right? So acetyl-CoA plus choline, they are basically there is a choline acetyl transferase. So acetylcholine is formed with the help of this transferase. So choline transporter makes them enter into the cell, and with acetyl-CoA it form acetylcholine. Okay, now this acetylcholine is to be packed into a vesicle. So here VAT help. VAT, which is vesicle associated transporter. Now with the help of VAT, vesicle associated transporter, this acetylcholine enters into the vesicle and packed here. Okay, so that's how vesicles are packed with neurotransmitter. Then thing come is how these vesicles come in contact with the membrane and release acetylcholine. That is next question. So that comes under synaptic vesicle pool or vesicle cycle. Okay, so what happens basically? This is a image which is explaining everything at one place. So what happens? These neurotransmitters are packed as here. This is a packed vessel, right? Now these are the pool of all the vesicles present over there. Now every pool is not free to release the vesicles toward the membrane. These are bound pool, okay, bound with the help of some proteins. Now some pools need basically calcium, some vesicles need calcium to release neurotransmitter, and then to release some vesicle as free vesicles which can come in contact and again repeat the cycle. Synaptic vesicles are basically 80% are present in reserve pool. Okay, synapsin is the protein which bind and keep them in pool, and 20% are readily releasable vesicles. Okay, fine. Then basically what happens, how these vesicles come in contact and release the neurotransmitters. There is a snare complex protein. Okay, what is snare complex? There are V snare proteins and T snare proteins. V snare proteins are basically synaptobremin, or T snare proteins are syntaxin and SNAP25. So these are the proteins which are helping for exocytosis of neurotransmitters. So how? For example, this is synaptobremin which is present over presynaptic terminal. This is presynaptic terminal, okay, and this is postsynaptic terminal, and we have SNAP25 and syntaxin present over there. Okay, the same arrangement is present on this side as well, right? So what happens basically when presynaptic terminal and postsynaptic, they come in close contact, these proteins basically twist in each other. So they are going to twist in each other, and that's why when they twist around each other and stretches this membrane, so this membrane rupture from here in middle. Okay, we have same arrangement here and we have same arrangement here. So both are twisting with each other and stretching this membrane which is present at center. Let's come to vesicle cycle again. So what happens basically, these neurotransmitters are packed in the vesicle having a bound form vesicles, and some vesicles are released as a free pool, okay, releasable vesicles. And what happens then, after that there is docking happen. Docking is nothing but the vesicle come near to membrane. Priming is when the vesicle attaches to the membrane, and then because of calcium entry present over here, the snare complex help in exocytosis of neurotransmitters. So that's how basically neurotransmitter is released in synaptic cleft. Then these are recycled to form again packing part of the vesicles, or they can make some endosomes, various empty vesicles. They fuse with each other and form endosome arrangement, which are recycled to form again a separate vesicles where neurotransmitter is packed in it. So these are the events at presynaptic terminal. Now coming to next part, which is events at postsynaptic membrane. Okay, so at presynaptic membrane the acetylcholine is released over here. Then what happens, there are some receptors which are ligand gated channels. So what that mean, when ligand, which is acetylcholine here, is going to bind on this channel, which help in conformation change of this channel and make a pore in this, so that all positive charge present over here, it can enter into the cell. Okay, so you can perform the answer like this, making a flow chart with diagram. So acetylcholine bind to acetylcholine receptors, which is nicotinic type of the receptor. Binding occur conformation change leading to opening of the channel. So sodium influx occur. We have sodium outside available here. So sodium from higher concentration to lower concentration, it will move. So sodium influx occur. Whenever sodium influx occur, the membrane becomes bit positive. We call them the end plate depolarization. So because of the positive charge entering here, which is causing miniature end plate potentials, okay, because of spontaneous acetylcholine releases. So whenever the positive charge reaches the threshold of these voltage gated sodium channel, so whenever threshold voltage of the channel reaches, there is opening of these sodium channels. So whenever these sodium channel open, entry of huge number of positive charge, that is sodium ions entry occur. So which is nothing but a depolarization. Okay, sudden positive influx is nothing but depolarization, but above threshold, mind it. Okay, if you want action potential video, you can comment below here. So whenever action potential, obviously what we are reading here, that action potential in nerve is transmitting to the muscle. So what happened here, acetylcholine bound to the channel, causing some cationic entry which reaches the threshold of the channel and ultimately leading to huge number of positive charge, which is nothing but depolarization. So ultimately depolarization of muscle happened. So action potential in muscle happened, which is again obviously starting all the muscle contraction events, which we'll see in next video when we'll understand skeletal muscle. So this is all about event at postsynaptic membrane. So presynaptic membrane and postsynaptic membrane, when you explain both these topic, it completes the neuromuscular transmission. So I hope this video makes you clear that what is neuromuscular junction and neuromuscular transmission separately. So if both are asked, how you have to answer this question, you can see these notes and you can explain it very well. You can score very good marks, mind it, very good marks. Okay, so this is all about neuromuscular transmission. I hope the topic is clear to you. If you have any query, you can comment below. In next video, we'll see applied part of neuromuscular junction. Thanks for watching, guys. Please subscribe if it's helpful. Thank you so much. Bye bye.